Today, we are going to present the result of our research project entitled Effect of the Type of Dishwashing Liquid in the Microorganism Viability in Kitchen Sponge. The kitchen utensils that we use to wash the dishes are causing problems for human health. Microorganisms can cause the major health problems, and these are going on kitchen sponge because due to their physical properties and can store large food remains that accompanied with humidity and temperature, and that is become an excellent medium for growth of these. So on the other hand, liquid kitchen soaps offer regular and antibacterial options to try to mitigate the effect explained above in the sponge. So for that reason, the objective of this study was to evaluate the antimicrobial properties of two types of these liquid soaps, one regular and one antimicrobial, over the tiny kitchen sponge. To observe the amount of bacteria that remain attached to the kitchen sponge, three strains for the Centennial College Culture Collection were used: E. coli, Salmonella, and Staphylococcus aureus. These bacteria were introduced in a sterile bag with phosphorus buffer and new 2 per 2 centimeter the kitchen sponge. Dilution was made and sprayed in TSA plate and then incubate at 35 degrees for 24 hours. An antimicrobial susceptibility test of liquid soap was performed. By means of three bacteria mentioned above, a mixture was made with phosphate buffer, and 1 milliliter of this solution was added to MSA in duplicate. And this was done, different dilution of liquid soap were added as shown in the image. And for the control, only phosphate buffer was added and they were incubated at 35 degrees for 24 hours. And two tests were carried out, one for the normal liquid soap and the other for the antibacterial liquid soap. Two new kitchen sponges were used independently to wash dishes for 3 weeks. One sponge was used with common liquid soap and the other with antibacterial liquid soap. Um, Once a week, the kitchen sponges were analyzed. The sponges were placed in phosphate buffer and mixed. Then cereal dilution were made and sprayed in TSA and incubated at 35 degrees for 24 hours. After this, a colony-econ was performed to follow a gram staining to see what type of bacteria had been isolated. And then a KOH test was carried out to rectify the result of the gram staining. And with this result, an oxidase test was carried out in order to know what type of method should be used and to identify the microorganisms. In this case, we use rapid staph and rapid one from positive and negative gram. And a new kitchen sponge was used and negative control.
Well, for the results, first of all, we have to say that for the bacterial recovery, the three tested strains, or the three most important strains that were E. coli, Yeast aureus, and Salmonella, three of them were recovered, being the E. coli, the highest one with 98% of recovery, Yeast aureus 89% of recovery, and Salmonella 81% of recovery. So the methodology used in this experimentation is good for the results that we are looking for. The susceptibility tests show us that in both a regular liquid soap and antimicrobial liquid soap, where clear zones that get bigger when the concentration of the soap increases. Also, we can see that it is a little bit bigger in the antimicrobial soap, but is not completely clear, so it's probably more than an inhibitory effect, like a displacement of the bacteria in the sample. For the sponge bacterial growth, we find that in both treatments, regular and antimicrobial liquid soap, the bacterial concentration increases as the sponge is used, being the week 3 the one with higher bacterial concentration, as you can see in the place shown in the figure 2. Something that surprised us is that the antimicrobial treatment did not show an expected inhibitory effect in the bacterial growth, but like regular treatment, the microbial concentration increased with its use. This is because soap does not kill germs. It mechanically removes them by attaching to the organic compounds in the bacterial surface. Also, the antibacterial molecules used in this type of dishwasher soaps are triclocarban and triclosan. These molecules need a high concentration to kill the bacteria, but the concentrations used in these soaps are usually low or too low to show an effect. And what this can generate is that it will develop the new bacteria that are resistant to this type of antimicrobial agents. After incubation and isolation, 13 different morphotypes were obtained, being 7-gram negative and 6-gram positive. 3 out of these 6-gram positive bacteria were oxidase positive, and after the use of rapid staph identification test, we obtained that these bacteria had a greater degree of resemblance to bacteria associated with cross-contamination associated with animals and humans, such as Cereus, Ehrlichia, and Scandinavium. On the other hand, 2 out of 7-gram positive bacteria were oxidase negative. The rapid one test results suggest that these bacteria are strains of Salmonella and E. coli, bacteria that you know is associated with foodborne disease. It is surprising that, despite of the use of soap and soap with antimicrobial component, this is not enough to remove the presence of pathogens in the sponges with which food utensils are cleaned. Therefore, a greater hygiene measure is necessary to protect our community for these foodborne diseases. Well, as a conclusion, we can say that there was no difference observed between treatments, meaning that they use antimicrobial liquid soap for the cleaning of the dish and plates doesn't have an extra effect in the bacteria concentration in the sponges and therefore in the plates. Harmful bacteria strains were recovered from the original samples after treatment, so it can represent a risk to public health if they are constantly exposed to antimicrobial compounds in the antimicrobial liquid soap that doesn't have the minimal bactericidal concentration. As I said before, it can generate resistant bacteria. And finally, it is recommended to use regular dishwasher liquid, as I said before, to avoid this mutant resistant bacteria generation, and regularly change the sponge to keep the bacterial concentration and at the appearance of possible pathogens at minimum. Because we saw also strains of pathogen bacteria in our samples. So to avoid the disease or the proliferation of the disease, the best idea is to get rid of the sponge before the concentration of the harmful microorganisms get enough numbers to cause an health issue in the person that is cleaning the dishes or eating in the dishes that are cleaned with that sponge. So that's all. Thank you for your time, and we hope you like our experiment or topic.